Information processing involves interacting groups of neurons, various neurotransmitters that encode information as action potential frequency. This is the last section of Chapter 11. It begins by talking about regulatory neurons, and these neurons will either facilitate or inhibit the activities of a presynaptic neuron. They may affect the cell-body membrane or alter the sensitivity of axon terminals. And you can see here some regulatory neurons synapsing with other neurons. Neurotransmitters are involved. There are over 100 neurotransmitters, and each one works differently. They may have a direct effect on the ion channels, or they may have indirect effects, and this would involve a G protein. G proteins are a diverse family of enzyme complexes. So a neurotransmitter bound to a G-coupled membrane receptor would act as a first messenger. And the first messenger, this would then trigger the release of a second messenger, which would alter the conditions or activity of the postsynaptic cell. So rather than opening or closing an ion channel, it will bind to the G protein and act as a first messenger. And we'll see examples of this. So here are some of the neurotransmitters you need to be comfortable with. There are only 6 here. Obviously, you don't need to know chemical structure. You don't need to know mechanism of action. You don't need to know location, but you should know the comments. So acetylcholine, it's widespread in the CNS and PNS. It's the most studied of the neurotransmitters. It is the one at every single neuromuscular junction. Norepinephrine is involved in attention and consciousness. Epinephrine is excitatory along autonomic pathways, that is automatic. Serotonin is important in emotional states and mood. Glutamate is important in memory, and it is the most important excitatory neurotransmitter in the brain. GABA is the most important inhibitory neurotransmitter. So remember that glutamate is excitatory and GABA is inhibitory. Oh, but I did want to talk a little bit about the G proteins. This, you don't need to know the individual neurotransmitter and their mechanism of action, but acetylcholine binds to chemically gated ion channels. Norepinephrine and epinephrine have indirect effects. They bind to the G protein and have a second messenger. So you'll see quite a few of these. G protein, second messenger. G protein, second messenger. And glutamate also has some direct effects. It opens calcium and sodium ion channels. So interpreting the information is really a processing through a variety of means. The information is translated into action potentials, and the messages are often interpreted just on the frequency of the arriving action potentials. For example, muscle contractions change in response to increasing frequency of action potential. So this is an example of just the frequency of the action potential. You can see a twitch. If you look at the graph down below here, where we have frequency of action potential, 20 action potentials per second result in a twitch. 50 action potentials per second result in incomplete tetanus, and 100 action potentials per second result in tetanus. The neurotransmitters released at a synapse may either have excitatory or inhibitory effects. The effects of the axon's initial segment reflects a summation of the stimuli arriving at any moment. The frequency of action potentials generated is an indication of the degree of sustained depolarization at that axon hillock. Neurons may be facilitated or inhibited by extracellular chemicals other than neurotransmitters. The response of a postsynaptic neuron to the activation of a presynaptic neuron can be altered by 1. The presence of chemicals that cause facilitation or inhibition at the synapse. 2. The activity underway at other synapses affecting the postsynaptic cell. And 3. Modification of the rate of neurotransmitter release through facilitation or inhibition by regulatory neurons. Information is relayed in the form of action potentials. In general, the degree of sensory stimulation or the strength of the motor response is proportional to the frequency of action potentials. And here are a few questions. The non-narrated PowerPoint has quite a few more questions at the end you might want to review. Which of these neurotransmitters generates IPSPs on the postsynaptic neuron? Inhibitory postsynaptic potential. GABA. Which of these occurs when a series of excitatory postsynaptic potentials reaches a postsynaptic cell, bringing it to threshold? Temporal summation. Which of the following events might lead to an inhibitory postsynaptic potential? Opening of potassium channels. Which type of channel involves the binding of acetylcholine to a receptor? Chemically gated. With a resting potential of -70 millivolts and a threshold of -60 millivolts, which of the following is a likely result of facilitation? -67 millivolts.